Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. We are covering a complete series with the name Python for Biological Data Analysis. Today we are going to cover chapter 3 and its name is Data Structures for Biology. Let me tell you, until 4, 5, or 6 chapters, we will covering these basic things, and after that, we will use all these basics for some exciting and interesting applications. So stay with me and keep watching and keep learning. So first of all, you have to open your web browser uh, and type colab.research.google.com, enter. Then when you see this window, click GitHub, then type here ASH 100. Once you enter it, you will see this kind of window. Click here, scroll down, Python for Life Science, click here. Now click here, chapter 3. You will have this kind of window. Click connect here. Okay. This is the course name, Learn Python for Biological Data Analysis. This is chapter 3. This is my brief introduction. And these are the course outline. We have already covered chapter 1, the foundation, chapter 2, the control structures. Today we are going to cover data structures for biology. And next we will cover all these chapters. Okay. So, okay, what are the learning objectives of this course or this chapter? In fact, the learning objective, we will talk about list and its application of list. We will talk about tuples and we will apply some dictionary stuffs and we will use all the examples from the biological data science or from the field of biology. Okay, so let's start. First, we are going to introduce or discuss or practice or learn lists and tuples. What are basically lists? Lists are the commonly used data structures in Python and it has some characteristics. For example, they will be ordered items. If you want to include some items in the list, so it will be in some specific order, and though that order will only be changed once we want to modify. Also, they are modifiable or changeable or mutatable, and it allows duplicates. For example, you have duplicate entry. For example, you have some data set of different genes, let's say. So maybe BRCA1 or BRCA1 gene is there, and you want to have two entries of BRCA1 in different rows or different columns, whatever you say, in your data. So list will be perfect because they allow different entries and they are heterogeneous. Heterogeneous means that list can contain integer like we talked in chapter 1. They can contain strings like a sentence. They can contain floats like decimal points. Okay, and they can contain boolean. Okay, so uh, this is a simple example. If you want to create a list, so you have to enclose it by this sign or by this, I will call it man bracket. So, for example, my empty list is equal to this. So it will be an empty list. And after that, if you see, if you print my list, let me make it bigger. Wait. Okay. So I wrote one, two, three, four, five different example I wrote here generally. Uh, Here I'm creating a list, my empty list, whatever the name you are using, and then followed by is the equal sign, and this is an empty list, just two brackets, and this is the print option, print, and then the name of my empty list, and it will be printed. And do not care about these things because we talked about in lecture 1, these are actually comments. Okay, now here is a list of integer. You can see number is equal to this one from 1 to 5, and then the list of strings, like it contains words. If you are confused that why there is no inverted commas and there are inverted commas, so we covered this in chapter first, that strings will always be in inverted commas, and integers and floats are free from the inverted commas. And this is a mixed type of list. I name it mixed list. It contains integer, comma, then in inverted commas a string, hello, then a float 3.14, and then a boolean. Okay, and then a print command. Uh, okay, and this is a nested list. Nested list means a list within the list. So a nested list then that contains integer, and then another list. You can see 3 and 4 is enclosed in another brackets, and then 5. So if we run it, let's see the results of all these scripts or commands. Okay, the first one was empty, the second one this list, the third one this one, the fourth this one, and the fifth this one. So all you can practice yourself just by creating an extra cell here and click the code. You will have an extra cell here, and just try and generating different lists. It will help you. Okay. Now we are coming to biology. For example, we are making a list in biology. So we have a DNA sequence, and I just wrote a shorter sequence, or the kind of ATG, CATG, and then I print that. And okay, once again, let me tell you, we covered this in chapter first, what this DNA sequence means, because we want that print command should print this variable DNA sequence, or this list, the DNA sequence, having this name or these words in the start. Then here we use some length function. Len, we used it, I think, in chapter 2 already. So we say that type len, and include len, then colon, and then calculate the length of this variable. The same way, we want to show the first nucleotide. So remember, if you want to show something in the list, because we are dealing the list, so the first one will be 0. So if you can see DNA sequence and then I mention 0, so it will always print the first. If you want to show the second one, that will be 1, and if you want to show this G, so that will be 2, because in the list the indexing starts from 0 and prints the last nucleotide and then the DNA sequence, but the last one will be always from the right side, -1. So let's run it. You can see this is the DNA sequence, the total length, this it worked, and the length is total 7. You can count, they are 7. First nucleotide is A, last nucleotide is G. You can try these things for yourself also. Okay, now we are talking some applications from the biology sides of these lists. So we can use these lists for DNA sequence analysis. For example, this is our gene sequence, and then we are interested for the counting of these nucleotides. So I will ask the program like nucleotide counts, and this will be ATGC, and then like I generated another kind of variable, and then I asked the program or the script to print this word nucleotide composition, and then for I'm using a loop, for loop. In chapter 2 we discussed for nucleotide, comma, counts in nucleotide counts items, and then also I'm interested in percentage, so I will use the formula of percentage. Percentage is equal to count obtained for a certain nucleotide divided by the total length and multiply with 100. And then I am interested to see the answer with one decimal point. We also discussed this in our last lecture that if you want to see the answer with one decimal or two decimal, and here is the percent sign. In last lecture there wasn't a percent sign, so let's run it. Now you can see the nucleotide composition A, the count is 3 and it's 30%. The T count is 3 and it's 30%, and so on. So also now we are making it more interesting, and we will include, let's say, pH measurements, this time points, this. For example, this is your data, and you measure different pHs of a sample. It was this, and then at certain points we got the data like 0, 1 up to 7. Now I'm interested in pH over time, and colon, this will be the word inserted in the print. And for I means in sequence we call it for a specific position, and here also for I, like this is I, this is I, this is I. Okay, also this is I in time point. So for I, time, comma, pH, this is for time. Sorry, this is for time and this is for pH. Enumerate time points and pH measurements. So pH measurements, this variable will be taken from here, and time points will be taken from here. And then we ask like print in formatted form hours, time, and pH. Calculate average pH. So the average pH formula we will insert. Average pH is equal to sum pH measurements divided by the total length of pH measurements. Okay, that will be average. And then I also discussed this n sign in our first lecture, that it generates another line. Okay, so let's print it. Now you can see we got the average pH and also a kind of a series of these things. Let me tell you, if you are a biology student and if you are thinking that this is so much complex, so don't worry, things are coming gradually. You will learn with examples, with you will learn in piece by piece. It is always impossible that you will watch my video and you will be an expert. But these video will for sure provide you a foundation, and if we will also do so many things in the next chapters. So at the end of this course, you will be able to understand something, to talk someone about the Python, to work somewhere, and if you receive an error, you will be somehow able, things will not be aliens for you. You will be somehow able to fix it. Also, nowadays, ChatGPT and other Gemini, all those things are available. So you can take help from them to generate your code. And but if you know something about code, that will help you a lot. Okay. Now I'm leaving this example for you because I will not cover all the example in today's session. It leads the sessions then longer. Okay, now the second thing is tuple. It's another fundamental data structure in Python, but the difference is lists were ordered. They are also ordered, but the difference is that they are immutable. Once created, you cannot change. Like list can be changed, and it also allows duplicates. It also allows heterogeneity, and they are a bit faster because of the immutable nature, and they use dictionary keys. What are dictionary keys? We will discuss later on. How we will create a tuple? You will create a tuple enclosed by the smaller parentheses. Remember, the larger parentheses were for list, and for tuple, the smaller parentheses. Once again, they are heterogeneous, so different types of floats and integers and booleans, etc., can be placed in a tuple. So once again, this is I'm creating an empty list here, then the print command, another tuple number tuple, the integer tuple, and then the string tuple. The only difference is that the parenthesis, and then a kind of mixed tuple. Now you understand why I call it mixed, and uh, then a tuple with a single item require trailing comma. So here we use a trailing comma in this tuple, so we can use it and without comma here. So let's print it. So here you can see one is with trailing comma, here single comma. It means you can also, if it's necessary, you can insert a comma. Okay. Now, basic tuple creation from biology codon. I'm using codon ATG, just name the codon and then print codon. Okay, and then if I'm interested in a composition, then sorry, it's not composition, position. So because in tuple you can also do or perform this kind of assignments. Let's say chromosome underscore positions. So, for example, we have a gene that is located on chromosome 12, and this is the position of that gene. So, you can use tuple to do this because tuple can maintain pairs of elements. So, now print gene located at chromosomes. This will be the print word, and got we have this kind of variable print position. So 0, as I told you, is the indexation. The print 0 means this one, and 1 mean this position. As or if you have some third category, let's say uh, position in a position, although there is not such word, but I just generated for you. So position in a position, that will be 2 here, the indexation. So the position will be index 1. This one is the position, and the chromosome number will be the position, the at 0. So you just click it. Now you can say, you can see, I just print also this, that they are immutable, so that's okay. I mentioned it already in the characteristics above. So gene located at position 12 and position chromosome 12 and this position. Now coming to some little more biological applications. So we are going to store some genetic coordinates using a tuple. For example, we have data of gene location, and that is now here we are creating a list because we are using this and a tuple inside. So you can see chromosome 1, chromosome 2, chromosome 3, and chromosome 1 again, because tuple and list both are able to manage or to accept the duplication. So that is fine. That's why chromosome 2 is 2 times. Okay, this is, let's say, one, these are the positions, this, and you can say this is the start position of a gene, and this is the ending position of a gene. And what is the plus and minus? These are the positive sense and antisense strands. Because DNA has two strands, positive and negative, or sense or antisense, whatever you call it. So let's say this is your data. Now we are going to print some gene locations and do something extra. So for I, as I told you, explains these terms I, and I. So for I, in chromosome, in Chrome, start, end, strand. So that means that I is actually, this one is chromosome, this one is the start position, this one is the end position, and this one is the strand. Now then, enumerate gene location. This is the variable name gene location. So we are going to enumerate this gene location, comma 1, and then length is equal to, okay, how to calculate the length? We ask the program, length is equal to, you will print the length, it will be end minus start. For example, a gene ends at 1000 and it start at 900, so its length will be 1000 minus 900, it means 100, the answer. So its length will be 100 base pair. So we ask the program to calculate this way and now print these information for us. So when you read it, when you play it, you can see the gene locations. This is the we asked to do like this. Now gene 1, that is chromosome, 1000 the start, 2000 this is the strand, length is 100 base pair. If you can see 2000 the end minus start, so 2000 minus 1000, so the 100 base pair. So that was all. I'm leaving the amino acid properties and sequence analysis practical exercise for you. Once again, let me remind you again, you just try to read and understand these scripts. If it's too bigger for you, you can read them in chunks. Let's say calculate GC content. So this is we just use a simple formula, nothing more. You don't need to be so much worried about. So for example, if you have a sequence and you want to calculate the length, you want to calculate the GC content, you want to calculate something else, so you can generate the apologies, you can generate those codes in pieces and then merge them together. So don't worry about the length of these codes. They are not so much difficult, they are easy. Okay, now we are coming to section 2 where we will discuss about dictionaries. And we will use these dictionaries for gene annotations and species data. So what are dictionaries? So dictionaries are powerful and flexible data structures in Python, and they also stores the value in pairs. And because of this property, they are more powerful. We can pair them different things, and we can trend them. We can create our data. We can manipulate our data, etc. Okay, so the key values are pairs, and they are once again just like list, they are mutatable. Tuples were not mutatable, and keys must be unique. In tuple and list, we saw that duplication is allowed, but in dictionaries, duplications are not allowed. And keys itself inside the dictionary, they are immutable, and values can be anything. So let's say, uh, for example, once again, how to generate a dictionary. So for list, we use a bigger brackets For tuple, we use the smaller brackets, but for dictionaries, we will use the curly brackets. So now I'm just creating an empty dictionary. My empty dictionary is equal to these curly brackets, then print command. Now I'm using some different integers and strings, and let's say so these are the student scores, and then I will print it. Here I'm using some mixed things. It contains names and sorry, name. This is Jane Doe, and age 30 is student false, scores maths and physics. So let's say I'm interested in a dictionary. So if you see, there are many items with one entry about this person whose name is Jane, his age is 30, is a student true or false, this Boolean, and what courses he is studying maths and physics. So in this case, as we mentioned that uh, dictionaries are able to keep the key value pairs. So dictionaries are important and powerful because with a single entity we can group different pairs, and then we will print information, and this is a comment, and then we are using the dictionary constructor. So let's see, uh, another dictionary is the name of the variable and dictionary. So for example, we are talking about some cars, brand Ford, model Mustang, year 964, print another dictionary. So let's say, okay, now you can see this was that empty code, this was the this one, and this list was here we asked and then we printed, and this one is the last one using the dictionary constructor. We did this. Okay. Now let's use one example from the biology. For example, we have a genetic code UUU, and then we pair it with UUU is a codon that translated into a phenylalanine PHE. So if you see, we grouped UUU, then colon, and then PHE, and here we use the comma. That means we are asking dictionary that these two things are pairs. This is paired, this is paired, this is paired. So the same way we generated this dictionary with the name genetic code, this item, and then we will print, and then for codon, amino acid and genetic code dot items, and then print the same thing. So you can see why it generated these kind of arrows, because we inserted it here. You can see here. Now this word genetic underscore code dot items, this items, this function. So we also use it above. So far we use some print functions, len function, enumerate functions. Today we are also using the item functions. So what actually is item function? Actually, it retrieves all the pairs from the dictionary name genetic code. So doesn't matter how many pairs do you have, what is the name of your variable, that doesn't matter. So that's why we use this item function in combination for the genetic code. So in your case, this genetic code word will be changed with your variable name. Okay. Okay, now because I'm taking much more longer time, so that's why once again you have to see to check all these examples by yourself. I will just give you a brief introduction. Now we are annotating some gene, let's say BRCA1, and this is all the list, the dictionary items. So you can see the protein length, associated diseases, function, and we generated each and every pair, like full name, its pair is this one, chromosome and its pair is 17, start this, end this, etc., etc. The same for TP53, the same for another gene CFTR, and then we we use some definition, and the definition was to display the gene information, and then we use some different trends option, and then finally we display it. So we will have this kind of thing. This is our result. So you can think on it. Because of the time, I cannot cover each and every example in a great and a sufficient detail. So this is the example from expression data, and you will be able now to read the code. Okay, this is from the species data management, and this is from the phenotype data of using the dictionaries, and this is from the sequence translation. If we want to write our own program and translate our own sequences, so we can. And here we provide test DNA. Otherwise, you can use some dedicated servers like Xpezi Translate. Okay, now unique identifier and sample collections. We are coming to sections 3. So sets, what are sets? So sets are actually unordered collection of unique items. If I make this word bold, unique, that means that sets also do not accepts the duplication. The item should be unique, and but they are mutatable, just like list we discussed in the start. And okay, what are the key characteristics? They are unordered, doesn't matter whether ordered or not. Unique elements, and they are mutatable. They are unindexed, just like in the list above, and we were accessing the content with those numbers, indexing numbers like 0 and 1 and 2 and -1. In this case, if you are dealing sets, so sets are unindexed and they are heterogeneous. Again, you can place different kind of data. Okay, and let's say creating assets. So how you will uh, create assets? Sets are created by placing all the elements inside a curly brackets separated by commas. However, if you want an empty set, you must use this set constructor as this create an empty set. So let's say this is my empty set, then I print it, print type empty set. So here I'm using a type function. So we will see the result and you will understand what it does. Okay. Uh, Number set, like from integer, then strings, and then mix it, and then my list with duplicates. So here I use some duplicates. Let's see. Okay, when we print it, so it generated an empty set, but in sorry, we are okay. Here we have a number and then print number set. So okay, it did not print this one. The thing is, it just print this kind of thing, class and set. The thing is, because we have a repetition here, and also again I use the repetition here, double four. So if you see, it has removed one of the four. Okay. Now, once again, let's do one cover one example, be specific from biology. For example, we have gene study one. and we have these four genes, and then gene study 2 we have again these four genes, and then study 1 genes, print it, and set operation. So we are in putting some different operations, intersection, gene study 1 and gene study 2 union, gene study 1 and then gene study 2. When we are generating a union, it will be like this. So if you don't know the intersection and union, these things are important for the person, people working in the kind of population genetics. They are generating some Venn diagrams, and those diagrams are actually using the intersection and union and all those things. And from there, we or the researchers comment that what are the two genes are intersecting or segregating in the same population or from the same location. So common genes, you can see that the intersection or the intersection says that two genes are common in the data. Um this one and this one, these are the all genes, and these two genes were unique. Unique means they are not common, and they were not present, or they are present only once. Okay. Then we can use these sets for ID management. You can once again cover this example. Then we can also use it for gene set analysis. You just need to read the code as much as you understand, and then uh, click this play button. You will have your answer. Okay. Now, and from clinical sample tracking, we are using this, and for the contamination detection, we are using this. Okay. So these are the different examples. This is a comprehensive example. I coined it. Um, Okay, so that was all for today. We discussed the lists, how to create, we discussed tuple, we discussed dictionaries, and we discussed sets. Four different things we discussed, and the summary of the takeaway is you have some idea about list, tuple, dictionaries, and sets. Mm, I received some emails from one of the viewer, and he says that the way I'm teaching is good, but I need to put more explanations. So, my dear, if I put more explanation and explaining pieces and chunks of those code, maybe in future we will continue, and I will also teach you some lectures, and I'll practice you some lectures that how to construct code. Uh, but apparently, if you believe me or not, these things are coming from the practice. The more you practice, the more you will learn. It's just like mathematics. And the second thing is, you will only practice if you have a passion for learning this subject or for learning Python. So that's it for today. If you are liking my work, please thumbs up and share this channel with your other colleagues. And nowadays, I'm busy with this Python thing, so the other videos are kind of disconnected. So once I see some interesting topic or thing or tutorial, I will bring it here for sure. Thank you so much. Stay happy. Bye bye.